we're looking to do is find the relationships at the molecular level, that is where a pharmaceutical agent can actually be delivered, or where a genetic modifier can actually be applied, that will impact the clinical side of things. So this is our great challenge, is how do we understand the link between this level of the nervous system and this level through all of these levels in a way that we can help further, first of all, our understanding, but also our ability to treat the diseases and the disorders. And that really involves integrating databases and models and tools to do multi-scale integration. So some of you may be familiar with an ancient parable, the story from the old Buddhist writings from the 13th century, that is really about blind men, blind monks, presented with an object in front of them, and they're climbing over this, describing pieces of what they're encountering. So this monk comes across the tail, but he feels it and he describes it. He says, this is a rope. This one comes across the trunk. He said, there's a python here. This is just like a python. Here, the tusks are the saber. I found a saber. The ears are a fan. The side of the elephant is a stone wall. And they're all describing in their individual terms what they encounter, what they see, what they feel, I should say. They're blind. But the challenge is that none of them individually recognizes that there's an elephant in front of them. And to a large extent, this is the challenge that we face in neuroscience. We have a tremendous number of labs all around the world measuring pieces of the brain, describing pieces of the brain, from subcellular levels, microarrays, EM, confocal, to cellular, to tissue, to whole brain scale. And they're using many, many different techniques across all of those levels. So we're producing data. There's no problem with producing data. And more of it is being produced every day. And the technology to acquire data about the brain is developing very, very rapidly. So it's becoming cheaper and cheaper to acquire whole brain scale data, tremendous amounts of data. There's the Allen Institute, which built a whole facility and acquired a gene expression for every gene in the mouse brain, in every region of the mouse brain. And they did it far faster and for less money than they thought would be possible. And there are more and more technologies, sequencing technologies, brain clearing technologies, optogenetics, that are making it possible to acquire and investigate the brain at larger and larger scales. And there are also new initiatives that are being announced, and it seems almost monthly that there's a new brain initiative being announced. Of course, there's the Allen Institute, which after they developed that gene expression atlas, they actually announced a new initiative to go in and focus on the mouse visual system. And they're going to systematically study every aspect of the mouse visual system, starting from the retina to the optic nerve to the thalamus to the cortex, categorizing all of the different cell types, all of their connections, all of their electrophysiological properties, their genetic properties, as well as their responses to stimuli, and then also the in vivo behaviors. So they will be characterizing in great, great detail over the next 10 years the full visual system of the mouse. And the billionaire Paul Allen, who's one of the co-founders of Microsoft, is funding that. The Human Brain Project was announced end of January this year. This is a European Union effort and I'll talk a lot more about it later in the second talk of this morning. But it's an effort funded by the EU on the order of €1 billion over 10 years to implement an infrastructure for modeling and simulating the nervous system. And that's something that certainly triggered a wave of interest in studying the brain. The BRAIN initiative then in the United States has emerged, also been announced earlier this year by President Obama. And there again, it was the order of 100 million per year being dedicated to studying and understanding the brain. President Obama made a statement that we don't understand, we've managed to understand the fundamental particles underlying physical reality. We've studied the most distant universes, but we haven't understood the 3 pounds, the 1.5 kilos of matter between our ears. And so we have a tremendous opportunity. There's a wave of interest. I know there are initiatives also in Japan developing, in China and elsewhere. And so there's a real opportunity globally to build upon all of these investments in neuroscience, but to get the whole world collaborating on this challenge. So I wanted to give a few examples of what I mean by data integration in neuroscience and multi-scale integration. And so, for example, you can imagine, let me see if I can get this to, I don't know, it's not going to play, but taking the morphology of a neuron, taking the 3D reconstruction, information about an individual neuron and its full three-dimensional structure and shape, taking data about the gene expression, which genes are being expressed in that individual cell, the electrophysiology, so characterizing how that cell fires, what are its electrical properties, how does it react to stimuli, and then building an electrical model, building a model that can predict how that neuron would fire given other types of input. And then running simulations, computer-based simulations, to evaluate and predict what would be the effect of manipulating one of these other effects. What happens if you change the morphology of a neuron? What happens if you change the gene expression or the distribution of ion channels, for example? How does that change the electrical behavior of an individual neuron? And of course, we know that in diseases and disorders, there are mutations in the morphologies. There are changes in distributions of ion channels. So this gives a tool to understand at the single cell level how mutations can affect the individual behavior of a single cell. Another aspect would be modeling a microcircuit, so a small circuit within the brain, about 0.5 millimeter wide, 1.5 millimeters, and that you can take all of those different models of individual neurons. If you've characterized the individual neurons, you can put them together and connect them in the same way that the neurons are connected in the brain, and actually run on a supercomputer large-scale simulation that helps you predict then the network dynamics. How does this circuit behave? And this, of course, is useful then for taking the same thing that I was talking about before, understanding the link between ion channel distributions, morphologies, gene expression, and linking it to how an individual brain circuit behaves. How does it change the computation performed in that brain circuit? How does it change the way that that circuit actually produces output and the dynamics there? And then you can imagine that as we're getting more and more data at the whole brain level, and we're getting more information about all the different cell types, their distributions throughout the brain, their electrophysiological characteristics, synaptic dynamics, connectivity, all the information about the circuit connectivity. There's more and more of these projects. There's the Human Connectome Project. There's many different connectome projects, brain areas, that then putting all of that together into a brain simulation, that you would then also have a tool to help you link the low level, the gene expression, to whole brain level activity and dynamics. And of course, that's where we want to go. That's one of the goals, is to be able to use computers as a tool for much better understanding the various substrates and the mechanisms that can affect whole brain behavior and dynamics. And finally, there's another example of an important aspect of data integration, which is multimodal brain atlasing. So traditionally, brain atlases were done by an anatomical expert who stained the brain, maybe in one or a few different ways, and they looked at it and they saw cell distributions and they saw folds in the brain, landmarks, and they said, this is this brain region, and they drew an outline. And so Brodmann was, of course, one of the famous anatomists who defined all of the Brodmann areas. And it turned out that those Brodmann areas actually corresponded to functional areas within the brain, which was what made it so successful and so exciting. But now we have an opportunity to bring in many layers of data into the same space. So we bring in not only the structural data, not only the distribution of where the cells are, or the ER are in the case of the Nissl stain, but also atlases of gene expression. So the Allen Institute has actually developed an atlas of gene expression in the human brain using they have 6 human brains that they've parcellated into regions and then computed the gene expression from. But also there are large-scale cytoarchitectural atlases of the brain. So for example, Katrin Amunds in Germany, along with Alan Evans in Canada, produced and released earlier this year one of the highest resolutions 3D atlases of a human brain, where they scanned an entire human brain at about a 10 micron, 20 micron resolution. And they're making all of that data available. And that gives us a picture of the cytoarchitecture of the brain. Where are all the cells distributed and the details there? But also myeloarchitectonics. So this is atlases showing where the myelin in the brain is, and so all the fiber tracts. And for example, using polarized light imaging that shows you the directionality of the myelin fibers, and going through systematically slice by slice through the brain, you can get a whole map that shows you where all the myelinated fibers go. Receptor maps also using autoradiography and PET imaging with specific ligands. You can get maps of the distributions of acetylcholine, dopamine, serotonin receptors throughout the whole brain. Connectivity. We know, as I mentioned before, there's the Human Connectome Project. There are many projects mapping out through brain imaging and otherwise, also through tracer injections, connectivity of different brains. So some of that's happening in the human, some of that's happening in the mouse. But there's a tremendous amount of raw data coming out about connectivity and resting state data. So in neuroimaging, one of the popular trends has been to look at what's called the default network, the resting state network of the human brain as a way of characterizing individual brains. And that's resting state data where the activity just kind of free flows without a specific task throughout the circuits of the brain. And then also functional mapping, where you're actually using specific tests, specific cognitive tests or tasks to highlight the activity in specific regions of the brain and looking at the variability there. Now what's interesting is that as we are standardizing the way that this data is described and being able to map it into common coordinate spaces, we're developing the ability to do what is really multimodal brain atlasing, where you can start to use these different layers of data to come up with new maps, new atlases, new ways of looking at the organization of the brain. And this is really something that we think it's at the threshold, it's starting to happen, but it's so important for moving beyond that very static architectural view of the brain to something that really integrates both structure and function to understanding the organization of an individual brain, but also the diversity of the brain organization. So maybe before I move on, are there any questions or comments? I kind of want to encourage as much interaction as possible. Has anybody any thoughts
please interrupt because and ask questions because this is not one way.
Yeah, I don't want this to be me just coming in and just.
Sean has flown all the way from Scandinavia to come over to see us, and I think it's important for us to ask questions. No questions are stupid questions. It might be interesting questions in his field that he may be able to answer. Even students are allowed to ask questions. No problem.
And encouraged.
And encouraged, yes. Any questions, please? There's so many people here from, I counted 6 different universities.
Great.
So some are big guns, some are small guys, so ask questions.
There's a question in the back there.
We have neurosurgeons, I know people from...
It's a great crowd.
Physicists, computer scientists. If you want to ask questions in Malaysia, I can translate for you, no problem.
And then, if you don't ask questions right now, you're going to have to interrupt me later. So, don't hesitate. Great.
Hello, Dr. Hill. I'm Eric. So you've shared with us a whole bunch of different kinds of data from the brain, from myelination to structural to activity maps. And I think one of the important questions is what kind of information? I'm more familiar with like calcium activity, so what kind of information, how do you know, how do you parse down which is important, which is not, because we know so little about them. How do you decide this in INCF?
So that's a really critical question, and I think, and it's not INCF's job to decide which is the important information, but in fact what we need to do is we need to have enough of that data brought together and able to analyze it and its relationship to specific behaviors or disorders, or to understand which levels of that are interesting and I think I need to turn off my phone. But that, I think it's a fundamental question. We don't have the answer. I don't think any scientist can say that, this is the answer. But by finding the relationship between that data and other data.
The approach right now is to just accumulate as much data and try to study the relationships.
So in the Human Brain Project, which is a separate effort from INCF, but in the Human Brain Project, the effort is to bring that data together, build models from it, and then by manipulating the models, you can decide, you can evaluate how much impact, how informative is the calcium signal for certain types of activity. Of course, we know it's a great correlative way of getting at the spiking activity. But what else is it showing? What is it able to reveal? Is something that we have to keep studying. So, other questions?
You can, people from physics, computer science, mathematics, even mathematicians here today, can ask any questions. Don't worry.
Right. But there's
At your center, you have that mega computer, all the computers in one room where you collect all the data, like that at the university?
No, actually, and that's something I'll talk about in the next talk, but the way that it's being structured is not to bring all the data together into one place, but to actually leave it distributed around the world and then take just the analysis and the pieces that you need from that data and then put it into a variety of supercomputers. So there won't be one single monolithic machine. Well, eventually there will be one in Germany, but there will be others around the world as well that can run pieces of the problem. So.
Any other questions?
So maybe I continue then, unless there's any others? Okay. So INCF, what is INCF? So it stands for the International Neuroinformatics Coordinating Facility, which is a mouthful. And INCF was really conceived out of the last Human Brain Project. So the Human Brain Project, the first one, took place in the United States, and it was during the 90s the decade of the brain that was declared in the US. Now, the Japanese declared the century of the brain starting in the 90s, so I think they had it right. But the decade of the brain with the Human Brain Project, the US put in $100 million into convincing neuroscientists as they're studying the brain to share their data and to put it into online databases. So it was kind of the first neuroinformatics project. And that's really where the term neuroinformatics was originated. Now, the problem that they quickly discovered was that, yes, they were able to pay neuroscientists to develop, measure data, put it into databases, and put it online. But you could not combine easily, you could not combine data from this database and this database and this database because everybody called the pieces that they were measuring differently. The data was gathered with different protocols. The data formats were all different. So you had this tremendous challenge in naming and in interoperability and in how do you, again, it's that how do you speak the same language for the field of neuroscience? And that's was basically, they discovered this is not just a problem for the US, this is a global problem, this is a global challenge. And we need an international organization that is going to help coordinate this at a global level, that is going to help bring together the community to make sure that we can speak the same language at many levels. And I'll explain some of those details. So, the Global Science Forum within the OECD, the Organization for Economic Cooperation and Development, saw this need. They brought in a lot of people from around the world to talk about this and to develop the plans. And it was in 2005 that the plans for INCF were endorsed by the Ministers of Research and then launched officially August 1st, 2005, with 7 founding members, including Japan, the United States, and other countries in Europe. The mission of INCF is to coordinate and foster international activities in neuroinformatics, to contribute to the development and maintenance of the infrastructure, the database and computational infrastructure worldwide for neuroscience, and to make everything that we do freely available. So when one country, invests and we develop some standard or some technology, we give it all away to all the other countries that are part of the INCF. So we make everything freely available. And that means making data available, but also making computational infrastructure available. And INCF, for example, was just on a grant where we were awarded a supercomputer in Stockholm. And this is something where we're putting some resource into that. We received a grant to get that supercomputing resource, but then we can make it available to the whole INCF community to use for their computational challenges. And then we also work very closely, and I personally work very closely with governments worldwide, funding agencies, for example, policymakers, as well as in academia, the academic leaders, and industry, because we really see that it's important to have everybody engaged in this discussion and adopting standards and communicating about what the challenges they're facing are and bringing the whole community together. So the INCF has a secretariat, the headquarters, so to speak, in Stockholm, Sweden. It was founded in 2006. It's actually hosted, although it is an independent and unofficially, it's an international organization, but it's sitting at the Karolinska Institute. There are 15 staff, full-time staff currently, but other additional contractors. And it is responsible for coordinating the scientific programs of INCF and the node activities, and I'll explain what that means. It manages the infrastructure, which I'll also explain. And then we have an annual congress that actually moves around the globe on neuroinformatics. So we've had this congress, it started off in Stockholm, we've had it in Pilsen in the Czech Republic. We've had it in Boston, in Kobe, Japan. We've basically had it go around the globe. Back at this year, it was in Stockholm again. But then we're looking at maybe in Australia the year after next. So we're really bringing this around the world to try to get as many people engaged as possible. And there are currently 17 INCF member countries. Japan, South Korea, India. Now, most of Europe and the United States. Canada is I hear, coming close to joining. I'm going to China after this trip and talking with a number of labs there. And we just recently had Victoria, Australia join this year. So this was a case where we were able to have just one region join. Unfortunately, we didn't get the whole country this time. But what they're doing is they're working now, they're using that as the seed to then get the whole country engaged. And it looks like they're on track for in the next year or two that the rest of Australia will join. So the INCF programs are really the core activities of INCF. And the way that the programs work is they're really triggered by community interest, by community activities, and suggestions to say, here's a challenge that we're facing in terms of data sharing, in terms of services or guidelines that we need, standards, something that is a driving need. So for example, we had a multimodal brain atlasing workshop earlier this year in conjunction with the Organization for Human Brain Mapping meeting, where we brought together experts from around the world to discuss what is it that will make it possible to integrate different types of brain imaging data into a common framework in order to accelerate our understanding of the organization of the brain. And they clearly saw us as the organization that would really provide that standardization and that community forum for agreeing on standards for doing that. So the community workshop is really a key activity. We pay to bring together experts from everywhere in the world to sit down and give us specific recommendations. So out of every workshop comes a report, a written report that describes what happened at the workshop, but also includes a synthesis of what was discussed, and what are the specific expert recommendations for INCF to pursue. So those reports are all freely available, and those recommendations go to the INCF governing board. So every node, every member country has a node, and every member country provides representatives to the governing board. And the governing board makes the decisions about which programs to fund, which activities to fund, and they take, very often, the output of those workshops are the core material for deciding when to launch a program and what should its priorities be, what are the key recommendations that should be pursued. And they define an oversight committee which includes professors, experts from the INCF community to define the specific objectives and task forces. And those task forces then are composed, they can be anybody from anywhere in the world, but of scientists that are working, it can be postdocs, PhDs, and usually postdocs or higher, that are all working on a specific objective. So they're working on developing a standard, they're working on developing a tool, service, and those are volunteers that are all together because it's in their interest to make this happen. And if that's not sufficient, if they need more resources to actually accomplish their objectives, INCF can award contracts to get these things developed. So targeted contracts to make sure that we develop these tools and resources as quickly as possible. So currently there are nearly 200 scientists engaged in the various programs. There's digital brain atlasing, multi-scale modeling, ontologies of neural structures, and standards for data sharing. And I'll go through quickly what some of these programs have delivered. If I can. So the program on digital brain atlasing has delivered a number of core products. One is something called the Scalable Brain Atlas, which is it's a web service essentially for embedding in your website maps of the brain and anchoring data on those brains. So it has a whole collection of atlases across many different species that can be embedded, and then you can also use the web API to anchor your data to points in that space. And it gives you a tool then for getting thumbnails, using a common vocabulary, and showing those brain regions where your data is linked to. The Voxholm space has been a very important development that actually brought together experts from the Allen Institute and other mouse brain imaging groups to develop a standard coordinate space, a reference space for the mouse brain. So for all the data that's being generated on the mouse, specific species, this C57 black 6 mouse, there's a standard coordinate space, and that became the Voxholm space. And what that meant was that then you had a community-agreed space into which all the other mouse brain atlases could be mapped. So that if your data in one lab has been mapped to Paxinos Watson, then you could actually translate that into the Voxholm space, and then through that space to another space like the Allen Institute's atlas. So it gives a very powerful tool. And to accompany that, we built the digital atlasing infrastructure, which is a set of web services for doing those transformations. So if data is mapped into one atlas, you can actually transform it to any other through the Voxholm space and through those web services. And that really gives the framework then for doing data integration of space. Now, the INCF program on ontologies of neural structures has done a whole range of work on developing standardized naming and classification. And again, not by INCF itself declaring that this is the only way to name these things, but by bringing together the community to say, here is a community consensus on a standard way of naming these things so that we could have agreed upon vocabularies. And one aspect of that, which is a tremendous challenge, is describing the different types of neurons in the brain. And so one project has been to develop the neuron registry. And the neuron registry has been led by Gordon Shepard from Yale University. And what he did is he contacted experts from around the world, the experts in specific types of neurons. And he got them to complete in this Neurolex, this online semantic wiki, the Neurolex, he had them fill out in a standardized form the properties of those neurons that all of these experts are studying. So we got a very standardized description of the neurons types, what they're connected to, their synaptic properties, and so on. And that's been curated and put into the neuron registry. And there's an effort to launch an Adopt a Neuron campaign where we'll get other experts to say, this is my neuron, I'm going to take personal responsibility for making sure it's fully characterized in this standardized way. The Neurolex wiki is, it's at neurolex.org, and it contains many different ontologies that have already been developed for neuroscience. It's also been used as a tool for curating other ontologies for brain regions and other things, diseases, molecules, and so on. And it's a public resource. The pan-mammalian reference structures has been developed in this group, and this is a set of reference structures across species, across mammalian species, that have been standardized upon. The Voxholm space delineations and definitions, so within that standard coordinate space you have the parcellations of the brain that have been named, and then the common upper mammalian brain ontology, which is an ontology for linking the common aspects of mammals and mammalian brain structure. At any point, don't hesitate to interrupt or ask questions. The program on multi-scale modeling has developed a model description language, so for describing neurons and synapses and networks, that is independent of the simulator that actually solves it. So that's 9ML, and that allows, so previously if you developed a model, it would be implemented on one simulator, and if you wanted to take that model and try it on somebody else's simulator, you couldn't. You'd have to re-implement the model using that simulator's model description language. And now this is an agreed upon, and again, a community developed model description language that allows models to be used in Neuron Nest and exchanged with NeuroML and basically interoperates with many different simulators, Brian, and so on. Connection set algebra is a way of describing connectivity in large-scale networks that's very general and very flexible, and then there's a library to allow that so that you could describe the connectivity of a large-scale neural network in a way that could actually be used, again, in different simulators. You only have to describe it once in this very general language, and it can be reused. And the multi-simulation coordinator allows coupling of different types of simulators. So if you're using one simulator that's specialized, say, in subcellular models, or one that's specialized in very detailed models of neurons, or one that is specialized in large-scale networks of very simplified networks, this provides an API for coupling those simulations at runtime so that you can use different simulators engaged in the same overall model simulation. And then the computational neuroscience ontology is essentially a way of naming and categorizing computational models so that people can easily find different types of models such as Hodgkin-Huxley, integrate and fire, but with a standard naming scheme and a way of essentially annotating and tagging those models. So the program on standards for data sharing has two task forces. One is the electrophysiology task force, which focuses on how do you acquire electrophysiology data from individual cell, single cell intracellular recordings to extracellular recordings to EEG, for example, and actually use a standardized format, capture the metadata, and work with industry vendors to standardize how their equipment also captures and stores that data. And then similarly for the neuroimaging task force, they're focusing on neuroimaging aspects, but that's primarily focused on human neuroimaging and the tools for sharing, anonymizing and sharing human brain data, as well as standard metadata, standard data formats. But then this important aspect, which is standard methods for workflow and provenance tracking. And this is something I'll explain in a bit more detail. But this is something that's essential if you want to work towards getting reproducible results, where in the end, you're ending up with a publication and a figure in a publication. And for somebody to really be able to reproduce that figure and understand all that went into producing that statistically significant result, you need to be able to track through all of the details of how was that acquired, which scanner, what was the protocol, what were the details of the acquisition parameters, how was the raw data processed then and analyzed in the workflows, how was that derived data actually then analyzed to produce that figure. And being able to keep track of all of that information is really key to reproducing results. So the neuroimaging task force, I'll go into a little more detail. This is the process that I was just talking about of tracking the provenance across all of those steps. And they're focused on developing a common API and a set of services for making that possible. And so a couple of the tools that they've developed as part of that process is, first of all, the challenge of how do you share human brain imaging data? It's too hard. It wasn't obvious. People would say, you know, sure, I'm willing to share my data, but there are some challenges there. I can't share brain imaging data that's personally identified, and it's not easy to know exactly how do I de-identify it and where do I put it. And so what they did is, again, it was this community of experts sitting down and saying, well, here's what we need to do. We need to write a script that will actually go in and it will strip out the metadata that's personally identifying from the DICOM images. It will deface the image, so it will actually strip off the face. And that all will be stored in an online database that's accessible to the community. And the benefit then is that if you submit your data, you can actually get automated QA or quality assurance measures back on your data. So if you submit it, you get back some result that gives you a QC measure, and you get an email notification. So this tool, this script, is available at xnat.incf.org, and it's essentially it's a big pool, a growing pool of community-donated brain images. Let's see. Oh, sorry. There's some next steps I can show you there. If I can navigate my presentation. Okay, so then the next steps, they're working on promoting it, extending it to structural and diffusion data sets, improving the user interface. So a lot of this has been happening, and then support for the Nifty format. And then also, they really want to build up a very, very large set across different types of scanners so that you can get QC measures specific to your setups, and also different populations of individuals as well, where the variability is very important to understand. And then there's also the challenge of, in neuroimaging, how do you find the data and how do you access it so that you can actually make use of it? And so they've been developing what's called the NI-API. So it's a web service API that allows you to actually query a standardized data model describing the neuroimaging data, so that you can do queries and find the populations you're looking for, and then set up a service to query off of that XNAT database. So again, at xnat.incf.org, it gives a way of making the queries on the metadata, finding the populations. If you want this gender with these properties from this locality around the world, you can query it through that API. And then they're working on making sure that all of the terminology and all the definitions of the metadata are in that Neurolex, that standardized vocabulary in the semantic wiki. And then they're working on implementing it and comparing that against some of these other large-scale neuroimaging databases that are available. And then they're also developing extensions and ways of building new data formats. As new types of brain imaging data become available, they're extending and developing new file formats to support that. And so they're working with, there's a set of free data available called the 1,000 functional connectomes, and they're using that data set to basically test out a lot of these extensions. And then the provenance tracking. This is what I was talking about before, where if you want to keep track of all the steps that you did in producing a figure, the challenge is that you don't necessarily remember what you did. And maybe the postdoc who did it or the PhD student that did it left already. So how do you reproduce that result? And so this is, what they've done is developed an API and a web service so that you embed in your analysis the calls to this web service that keep track of every step you take. So when you start working with a data set, you send a little message. You say, I started working with this data set, and you reference that data set. I applied this analysis, this version of this analysis script at this stage. The output was this file. Every single step is tracked so that it basically, and it uses a W3C standard, so there's a W3C provenance model available now as a recommendation. So this is the organization that monitors the standards for the World Wide Web, and this then becomes a standardized description of the provenance. So this is something where they have prototypes of this. It's not yet fully production ready, but if there are people interested, they would love to engage with you. And so they're working on integrating that with the LONI pipeline, which is at UCLA, and that's of course very widely used, and NiPipe, which is a Python neuroimaging pipeline. And then what's great about this task force is actually the tool developers are sitting on it. So the people who developed FreeSurfer and SPM and so forth are on that task force, so they can very quickly take those standards and take those ideas and implement them into the tools directly. So other activities at INCF are in the area of training. So there are actually opportunities there to support new types of courses. And the idea is really that INCF, they don't want to get into the business of funding every year a given course, but rather triggering something new to get enough support that it could become self-sustaining. And there have been a number of in 2012, there was an Allen Brain Atlas hackathon. There was an advanced statistical modeling of neuronal data, modeling across scales. And then this year, there was actually a course in Havana, Cuba, a short course in Germany, and in Belgium. These come in, basically, they get proposals from all around the world. There's a committee that evaluates them, and then they provide funding to make those happen. In addition, the Google Summer of Code is something where INCF acts as the coordinator. We collect project ideas from the whole community, things that they would let, that the community says, hey, this would be really great to have developed, but we need somebody who could actually have the time to work on it and get some payment to actually develop that. And what's fantastic is Google will actually pay those students to do the work over the course of a summer. And so we basically match up the mentors, the people who want to get those projects done, with the students that are available to accomplish those goals, and then they get paid actually through us, but Google pays us. So it gives us a way to get people engaged directly. Okay, so any questions on that section? Anybody?
Any questions? Any questions? Silence on that?
All right, everything was perfect. Yeah, good.
What are some examples of projects that were done in Google?
Right. Some of the Google Summer of Code projects was, for example, some work on the neuroimaging pipeline, adding support for standard, so some of that provenance modeling work. Some of it was also on adding capabilities to our data federation. So again, cloud-based management of data. And then there have been tools for doing electrophysiology analysis. So there's a whole, it's very, very diverse actually. And it's really up to whatever type of tool, but it has to be, it has to be manageable within a 6-month time frame, but it's something very open. Yes.
Would that be another course on where do I check this?
So there's a training section of the website at INCF, and that would list it. There's also, as we tend to list courses, there's a section also that just lists training opportunities at INCF, and those are not necessarily the INCF sponsored courses, but any opportunity around the world. So I'm sure that there's a course that would be, yeah, INCF.org, and then I think it's at /training or training.inf.org.
I have one.
Yep.
You're going to discuss with us like some of those data sharing and so forth, how we can participate, what does it mean?
Yeah.
And also, do you have any like e-courses or anything like that?
That's actually something that I really emphasized as part of the training courses, that what we're trying to do, it hasn't happened every time, but for all of the training courses that we sponsor, we insist that we get all the presentations, and we also film those so that the material becomes available for others to use for their course. So that's something where I know some of the material is online at the training section. We're working on getting the rest, and there are also, we have a YouTube channel. So on YouTube, there are many, many of these lectures filmed and put up there for free. So again, everything that we can make available is up there, including slides. So, yeah. All right. I just need a little water, so bear with me.
I think we're breaking at about 10 something after this lecture for a short tea break, so don't worry, you guys will also get
All right. So I wanted to give a little bit of an idea of kind of the details of how is it that we can actually achieve data integration. And you know, I've been talking about this. There's lots and lots of different types of data, but there's also a lot of reasons to integrate it. And the fact is that just simply by putting together the data sometimes, it allows us to ask new questions. If they're in a common framework, we can actually start to just find relationships between the data more easily. But it's also extremely valuable to teach. If you've got access to the data and you've actually been able to bring together data, and you've got, for example, provenance information, which I was just talking about, imagine how powerful that is to teach your students how to go through an analysis task, how to follow through an analysis process, to find the data, right? If this is accessible and annotated, you can find the data you're looking for much more easily. Of course, to share it, to publish it, we're working with a number of publishers to really change the way that data is published, defining things like data articles that really just make the data available, to replicate experiments, to analyze, to visualize, to model, to simulate, and ultimately, of course, all of that is to understand. So a key piece, not the only piece, because of course there are many incentives and cultural issues to getting people to actually share their data, right? The culture of science has not been built around data sharing. Very often it's been around, you know, this is my data, I got it, you can't have it, I'm going to find all the insight I can find, you know. And so this is something we have to get out of. And then of course the big challenge there is the incentives, right? That you know, if your path to getting tenure is to acquire a particular data set and then get all of the high impact publications out of it that you can, you're not going to be very inclined to, you know, to share it unless you get some credit for it. Okay? So what we're building is a collaborative research infrastructure for neuroscience. And this is something where we need to make it easy to share data, to make it securely accessible, to keep the authorization so that you're sharing it just with the people that you think you are sharing it with, but also make it easy to collaborate, to publish data. So if you want to make data available, but you want people to be able to cite it, to say, hey, I used this data, which this individual contributed. We need to be able to keep track of that. Keep track of community ratings. Now, this is like on some, you know, if you go to a consumer website where you're going to buy products, there are all these reviews and ratings on Amazon or elsewhere where you get 1 to 5 stars, this is a great product or this is terrible. Well, the same thing could be said about data, right? You could have reviews to say, I tried to use this data, but actually it's full of missing values and it's, you know, it's really frustrating to use. Or this is absolutely beautiful data. We all know when we see beautiful data, right? To preserve data. Now, this is something where many groups have come to us and they said, you know, I don't want my data to disappear. I have a postdoc that was working on it, and that machine that they were working on, because they just kept it on the hard drive of that one machine that the postdoc used. And then when they left, well, that machine was taken and the disk was reformatted. And, you know. So we want to kind of we want some better data management policies in place. And a lot of times labs need help doing that, right? It's not so easy. It's not that everybody understands or has the resources to take advantage of that. So this infrastructure can provide a way of making it easier to keep track of different versions of data, to archiving it. If the data is really valuable and it's highly rated and a lot of people are using it, we can make sure that a copy of that data is kept. And also, if it's sitting on a slow server in one part of the world and another group, say in Malaysia, wants to access it, let's make sure that there's a copy closer to them. So in Singapore, for example, Amazon has a big server center. Let's make sure there's a copy over there so that it's much more readily accessed. To federate data, what does that mean? To federate data means that we're not going to move all the data in the world into one central repository. It's just not feasible. And nobody wants us to do that anyway. They want to keep control of their own data. If they see that something, they want it on, near them, somehow under their control. But what we want to do is we want to provide access to it in a standardized way. So it's easy. You can just log in and get access to data repositories all over the world. Federated search. That, again, means being able to search for the data you're looking for across the data repositories all around the world. So you don't have to go in and get a unique login for that repository and a login for that repository and so on. And then if you want to search, you have to go to each website and search for all of those. But give one tool for searching all of them together. So, the web solved this for websites. But the challenge is that we don't have the equivalent of the web for big data sets, in terms of really data management. So we also need it to be very easy to use. Now, the idea of Dropbox, I don't know how many of you use Dropbox, but it's so easy, right? You put it on and it replicates that folder on your hard drive with the cloud, and it's great. But it costs, and it can cost quite a lot if you start using a large amount of data. But it is very nice and easy to use. And what we want to do is provide sort of that Dropbox-like functionality, but for scientific data. And then you want a layer on top of this, and this is sort of like the document object model for the web, but we want it to actually get an object-based view of the data itself, right? So that you can know, how do I read in that data? How do I access the contents of that data in order to analyze it? Because if every data is in another format and you don't know anything about that structure of that data, you can't actually make use of it. With semantic annotation, so that you actually, now the web is transitioning to the semantic web technologies, where all the terms are defined in ontologies and you can find the links and the relationships between them. And all of that actually makes it possible to integrate data, access it, analyze it, and then track the provenance of it, right? So that we can give attribution, so that in the end, if somebody is using your data or your analysis script or your service, that you get some credit for it as well. So this is pretty ambitious, right? This is kind of crazy and want to do a lot, want to provide a lot. But I think it's very important. I would love to hear your feedback on pieces of that are missing or other things that should be thought about. But I don't know how many of you have heard of Jim Gray. Jim Gray is this guy, sailor. He actually was one of the researchers ultimately at Microsoft who led a lot of the developments, the technology developments, the ideas around managing big data. And so he was involved in a lot of projects that were handling large amounts of data and collaborative research infrastructures. And in fact, there's a story there because he went missing. he went out on a cruise on his boat, and people were really concerned because he didn't call back, he didn't call in, he was missing for a day or two, and they were worried, where did he go? And it was a lot of his friends in this community of big data analytics. And what they started to do, because he was missing for longer and longer, is they started networking and saying, hey, let's tap into this source of satellite imagery and this source of airplane photos, and let's bring all of that data together using his techniques, basically, to start doing a global search in this whole, because this vast region of the Pacific Ocean that he went out into. And they were looking, well, this is the trajectory he was on, and let's use all of the techniques he invented to do that analysis and try to find him. And unfortunately, they didn't succeed. But he contributed a tremendous amount to the community. And one of the things that he advised in undertaking these initiatives where you have a grand vision of what you want to deliver, you want to provide a collaborative research infrastructure, don't build the whole thing. Look for the critical, the core, the 20% that's going to make 100 times improvement in the lives of the users, and deliver working prototypes early and often. And in my mind, this is really key. We don't know the whole solution. And the whole solution depends on you, depends on the community, depends on others getting engaged. And in fact, there's an example in the cancer community. The National Cancer Institute in the United States, they put $300 million, I think, into developing a very comprehensive, all-encompassing, beautiful, geniusly engineered system for data integration and collaboration in cancer. It's called CA-BIG. And the challenge that they faced is that they weren't getting the users to start using it. Right? So they kept putting more money, saying, well, if we just make it better, then more people will use it. If we build it, they will come. And I think this is something that we have to really look out for, that the trick is building flexible, small pieces that can be adopted for multiple purposes, and make sure you get the users from the early point, so that the users can shape what it becomes, so that it becomes an integral part of everybody's life, not just going and building one comprehensive infrastructure that just does everything. So we started with a piece. And one piece that we see is an international federated data space, which is basically how do you manage data distributed in repositories all around the world. And then on top of that, there are different services. So the knowledge space semantic encyclopedia, I'll explain that. Analytic services, so where community members have specific types of analysis they can provide. Brain atlases, which I talked about earlier. We've got some of the services for transforming between brain atlases. And then a layer for accessing curated data and doing queries and tracking provenance. And then on top of that, then the community should be building their workflows, their analysis, their visualization, modeling, simulation, whatever it is they want. And there should be a great diversity of, this should really be the enabling layer to building different types of portals, specialized portals for finding specific types of data, doing analysis, doing things, for example, in neuroimaging, that's one type of a portal, but you may have other portals focused just on modeling and simulation. So this is really the core idea of the infrastructure. And with the first piece, which we wanted to address, just try to address really simple, basic challenges, which is that when a granting agency or somebody tells you you need to share your data or you want to share your data, right? How do you do it? It's not so obvious, especially when the data size becomes big. You can put it up on a website, but then you have to host the website, and you need to be, and then you point to an FTP site, which has the NATAR compressed file, and you don't have all the rich annotation of those files. So it's not necessarily easy to do. And then how can you share that data just with specific parties, right? Maybe you're not ready to make that data fully public yet, but you want to collaborate with somebody. And so you want to control who accesses it. And what about backing it up? if you want to make sure that there are replicas of that data. And also, is there a space where you can just go and look and say, has somebody already shared data about this before? And so we thought about how could we make it easier. So one piece is to make it as easy as a Dropbox, but for scientists. That means being able to drag and drop any data, text, or images, and to be able to control access to that data. So what we did is we actually did what INCF does, and we held a community workshop on the issue. And we brought together experts from around the world, from the San Diego Supercomputing Center, from CERN, so these are the physicists that are dealing with big data all the time, the Allen Institute that had been doing these large atlases, from RIKEN in Tokyo, from parallel supercomputing centers, from synchrotrons, other supercomputer centers, basically all over the place to come and tell us, give us their recommendations on how to build this core piece of the infrastructure. And they did. They wrote a report, and the report's available on the website, and it basically tells us, here's what you should do. You should build this. And so what we did is we said, okay, community told us what to do. We're going to now go and contract to get this done, because we didn't have the programmers in-house to develop it all, and we found an expert that we could contract, a company, small little company, and within 6 months we had the contract done, implemented, and everything was up and running. So it's in beta, I should say. It's still development, but it's working, it's running, and people are hooking up data servers to it, and it's really functioning, and that's the INCF data space. And this is something that supports, first of all, just making data repositories available worldwide. But it's a place where you can discover neuroscience data, you can make your data visible. So we also give away on our service 5 gigabytes of space for free if you have small data sets that you want to make available. But you can then set permissions on that to say, I just want to make this available just to this individual. And you can share your large data sets that way. And you can also send links, so anonymized links. If you just want to email a link to that data, you can send it through there. So it's a way of making data public, collaborating, and then also establishing, again, it gives you the tools for saying, if I want this data set here to be mirrored over here, it gives all of the infrastructure to do that. And we run this using the Amazon cloud, actually. So the data stays sitting on the data repositories around the world, but the Amazon cloud gives us the metadata infrastructure for keeping track of where that data sits, on which servers. So this is extremely scalable. This is something that can scale up to exabytes of data, because the data itself we're not storing, we're just pointing to where it sits, and it then gives the way of moving it around in high-speed parallel transfers. So it's keeping track of the location, the replication state, archiving the data, searching for the data, and it can be any type of data. I think I made this clear. So you can take, for example, a mixture, an experimentalist can take a mixture of electrophysiology traces, neuron reconstructions, models, staining images, and just use it as a distribution point to making it public. We also had a use case where there's a group in Italy doing these large-scale brain-cleared images. So these are multi-100 gigabyte images that they wanted to make available for another research group. And what they had been doing is taking the train with a hard drive to go and get it up there. But now, using this infrastructure, they could do a parallel transfer. So the provides ways of using multiple threads and doing high-speed transfers and just keeping that replicated and up-to-date in an ongoing way with that site. And then we had the case where we had Japan was concerned after the last earthquake and nuclear disaster and all of this. They were concerned that we would like to be able to mirror some of our things off of this island and make sure that there's a copy should there be something, again, that causes this issue. And so as part of the data space, we're getting them hooked up, and they'll be able then to do replication with other collaborating sites. So whoever is able to provide the storage, and we have a number of supercomputing sites hooking up where they have storage available, and they just need a collaboration agreement to replicate that data. Also, the Allen Institute, for example, we provide a European mirror. So in Japan, there's an Asian mirror in Okinawa of the Allen Institute data, but the data space can provide an infrastructure for synchronizing that data across these continents. So this is what it looks like. If you go to the website, to ids.incf.net, it finds the nearest site to you. So here I think it would go to IDS-AP, Asia Pacific. It would find it's the closest, fastest site. And then you log in with your INCF site, so you can get an account on the INCF portal. And then you can basically browse the federation of different servers. And now again, this is in beta, so we're still working on adding it. We haven't set up all the metadata searches yet, but you can already go in and browse different data repositories and go in and get data. For example, the brain images of the mouse that was used to do the Vox home space and the things I'd been talking to you about. You can get huge numbers of human images as well. And this provides a way to just get them, access them, download them through the web is one way. But you can also use, if you want to do high-speed transfers of large amounts of data, there's a Java client that actually does that and uses multiple threads. There's also, if you're a more sophisticated user, there's this whole set of command line tools that are Unix-like. So instead of doing ls or cp, you do ILS and ICP. And you can basically, I could pull up my machine here, and if I have an internet connection, just do ILS, and it will immediately show me a listing of the files in the data space. So it gives me a way then to access this whole virtual, this whole set of data repositories as though it's an attached local hard drive, essentially. So some of the next steps for the data spaces, we're working on really improving the web interface, making it possible to get previews of images and documents, make it possible to also use things like Amazon S3 and Google Drive as part of it. And there are Dropbox-like clients for the Mac, PC, Linux, and iOS. All of this is coming with, what we've done now is there's an open source Dropbox clone called OwnCloud. And OwnCloud, basically, if you want to set up your own Dropbox without paying for Dropbox, you can use OwnCloud. And what we did is we contracted the company that makes OwnCloud